It's all in the same
So
day.
you finish at 12, you come down here for something else and this?
Yeah, so we finish at 12, come down here for a tube from 2 to 4, and then we come here for classes.
It's a long day.
I was at Hawkesbury, but then we gate crashed the tutorial today, so we're here.
And he's let us stay in it, so we're like,
Yeah.
Okay, all right, cool. All right, shall we get busy? We said we'd start on the hour, didn't we?
What am I doing?
Green light's on, means you're being recorded. Anything you say might be used against you. So today we have kind of some heavy duty going to get through. And as I said and warned you at the beginning of the semester, this is a very conceptual unit, so you need to kind of stay with it. But when we finish amino acids this week at Hawkesbury, I'd recommend very strongly you go online and look at the guiding questions, which will come up after each topic, i.e. amino acids 1 and 2, and also the concept notes. A couple of things I just need to kind of relay to you before we get too busy. This unit, you see lots of chemical structures. Last week, we talked a little bit about one particular pathway. You'll notice I didn't use too many chemical structures. There are some you have to know, which are the major regulators of energy carrying within cells. We're going to see one today. For the rest of it, my focus is mostly on names, because typing in chemical structures and finding things is particularly difficult. So for the exam, there will be some chemical structures you need to know, and I will highlight them here through the lectures. So you need to know this chemical structure. The rest, well, you know glucose pretty well and a few others, but I will be not asking about chemical structure, how things move around in this unit. Is that kind of clear? It's names. You can type in a name and find information. Much harder to type in a chemical structure and find information, isn't it? Unless you're a chemist and you're more comfortable with it. That's the key thing. Now, because I said this is very conceptual, we're going to work through the models, and last week we started on amino acids. We saw mostly ingestion, do the same with fats, we'll do the same with carbohydrates, then absorption, getting it into cells and its behaviour in the cell, a bit of synthesis of a few little simple amino acids. Today, during the break, I'll talk a little bit about your lab program as well, how that relates more. But today we're getting more into what amino acids can be used to make other things, but also pathways like managing too many amino acids. And we'll get to that through today as well. But some of these are rather tricky, and I mentioned last week, allosteric enzymes are the ones we want to focus on. Those characters that get busy changing things. The executives saying move faster, move slower, depending on supply and demand, which today is part of that particular process. And I said I'd show this slide a few times during the semester, you'll get sick of it, but you need to highlight where we are. And today we're going to start off, just for now, over here, amino acids coming in. We saw proteins last week, we saw a bit more transamination last week, and we talked a bit about some of these characters, other molecules derived from amino acids. Some of them extracellular messengers, but also some of the components of those amino acids are being used to make other things for you. And we touched on some of those. We're going to break off from that fairly soon after and get down to this particular pathway and then head into the urea cycle. Why you have to urinate basically first thing in the morning. What happens when you have a high protein diet, for example. Now, if you've looked at the slides already, like I asked you to do, what I'm talking about is not so unfamiliar. So if you haven't looked at the slides, today is going to get pretty ugly pretty quickly. So that's why I'd strongly encourage you to look at the slides before you come in. Don't have to read them in detail, but have a look at them. So let's start off over here. What else can we do with these amino acids? Some of them, particularly. Here's a couple of key examples. And let's walk through these slowly. Tyrosine, what else can happen to it? Phosphorylation. Very useful amino acid. In my mind, probably one of the more important ones to deal with in terms of signal transduction. But tyrosine, with its ring structure, with this enzyme, the hydroxylase, some help of oxygen. This other character, our cofactor, what do we end up with? DOPA. Anybody know about DOPA? It's in your brain. It alters your brain function. It makes you feel a little bit good sometimes. It alters neuronal transfer and turnover. We talk a bit about an advanced cell. If we bring in this decarboxylase and our cofactor we saw last week, remember that guy, PLP, pyridoxal L-phosphate? What's it doing? Help to make this thing dopamine. You've heard of dopamine, I hope. Yes? Also a brain functioning, this case, amino acid derived molecule. It also makes you feel different in your neuronal cells. It's a messenger. It's a molecule that alters brain function. If we bring in this hydroxylase through ascorbate and some more oxygen, that dopamine can be used to make norepinephrine, aka noradrenaline. We take one more step down here with this transferase and another cofactor here, adenosine. We make adrenaline. Where do you make most of your adrenaline? The adrenal gland, the adrenal medulla. Where does that hang out? Where's your adrenal gland? Sorry? It is. It's just sitting on your kidney. I like that word, superior. It's sitting on your kidney, one on either side if you've got two kidneys. Around that adrenal gland is lots of fat. We're going to talk about that later on. That lots of fat is the last fat you will lose if you lose everything else that you have is fat. That fat's used to make things like steroids. Here's one that comes out of the adrenal medulla. That's directly linked to your brain, a neuronal collection from your brain straight down your spinal cord, straight to your adrenal gland, to the medulla, which we'll see later on as kind of whitish tissue, surrounded by brownish tissue cells. The whitish tissue, direct connection to the brain. The moment you're stressed, what do? You dump adrenaline out. Changes your blood flow, your heart rate, turnover of glucose. The tissue surrounding that medulla, called the cortex, produces cortisol, corticosterone, aldosterone. And when you're young, going through puberty, those first few years, making testosterone. So those two cell types are interlinked to what your brain's doing as well. Remember, adrenaline's a chronic stimulus, adrenaline's acute stimulus, cortisol's a chronic stimulus. Adrenaline stimulates like that, cortisol, transcriptional regulator. Another one, glutamate with PLP and this carboxylase makes GABA, another brain functioning cellular messenger. Down here we're seeing histidine with PLP again. God, that character gets around. Making histamine. Where do you find histamine? Who's doing immunology? Some cell biology, some basic physiology. You've all heard of antihistamines, haven't you? It's a neural functioning stimulator. Immune cells stimulate release of histamine and change your behaviour. That's where you often see allergies. Tryptophan, another complex one. So you'll notice these are relatively complex molecules using this hydroxylase. One step, the hydroxytryptophan, PLP comes in again, you make serotonin. Now that's a feel-good hormone in the brain. You might want to jot down tryptophan. Where do you find lots of tryptophan? In the stuff you eat from the sweet shop. You all eat chocolate, don't you? Chocolate makes you feel good. I know someone who eats this much chocolate every day. Just had liposuction, laparoscopy, sleeve, now is removing all the excess skin because a block of chocolate each day, and I mean a big block, made her feel good, but it didn't stop her eating. Changes your brain function. So there you are, all of your chocolate's going in your brain to make serotonin. That's why you feel good after you eat chocolate. So now metabolism is starting to affect brain function, not just what it does to your metabolism. Boy, that probably quietened everybody down. I'm going to go home and eat a bar of chocolate now, chocolate milk. Lots of things alter your behaviour or your metabolism. Can I move to the next one? Is that a yes? Are you still in shock? Back off the chocolate. Let's go and make something good for you. Let's start with succinyl-CoA. Now, I want you to draw a little circle around CoA. You're going to see CoA shortly. That's a chemical structure you need to know. You won't have to draw it. Let me make that clear. In the exam, you won't have to draw any chemical structures. You'll get one. You have to tell me who it is. Is that kind of clear? There's no drawing chemical structures in this unit. But there it is with our friend glycine. Relatively simple molecule, isn't it? Carboxy, amino, 1 carbon. What are we going to do? We're going to work through all of these steps to make who? What do we make down here? Heme. You have lots in your red blood cells, don't you? This is a biochemical pathway. It's taking glycine, an activated molecule, you're going to see succinyl a few times, activated because CoA is on it, through a whole series of steps you've activated, bring glycine in. What have you done? Connected glycine to the succinyl through this synthase. Out goes CoA. Just put a circle around the CoA, and I want you to note the SH group. We saw phosphate activate somebody. CoA does the same. It uses sulfur to do it. All those extra electrons you saw in chemistry? But what you've just done is recycled CoA sulfhydryl, so it can be used to do another job of activating another molecule. That's why we'll see it a lot during the semester. With this reaction, we end up with CO2 coming out. We end up with this thing, delta amino leuvolate. Okay, what's important about that? We are now potentially on the pathway to start bring all this together through 1, 2, 3, 4, 5, 6, 7 steps, and just circle the incorporation of the iron group, the Fe2+, to chelate that into the heme group. Now, is that hemoglobin, the protein, or is it just heme? It's just heme. You have to stick it in that big protein, a bit like BSA you're playing with in the lab. So you've just made that heme function, and you see a whole lot of steps in there. Now you'll notice the enzymes aren't involved there. Let's look down the side. There's the delta amino leuvolate. There's a bilirubin. We've got now phyrogen. We're going down to this one, and then this one, and this one, and this one. Aren't they ugly names? But each of those is a biochemical step your cells have to go through to make this function, and then put it in hemoglobin. Each one of those biochemical steps, we see there's a red cross. That red cross, if there's a defect, now this is critical for your report, if there's a defect in the gene, point mutations, SNP, whatever it is, maybe some other artifact, some kind of defect in the gene, that enzyme doesn't work, you end up with a specific disease, DOS porphyria. If this enzyme doesn't work to go down here from this particular substrate, down to the next one, position 2, what ends up happening? Acute intermittent porphyria. Those are blood disorders. They change the individual. There are a number of these diseases. They're the ones, the few that we check first when you come into the world to see that you don't have these diseases. What are they? Genetic disorders. What's your first report topic? Genetic disorders. Each of those enzymes contribute to a different type of disease state, simply because making heme is kind of critical, isn't it? Doesn't mean necessarily these individuals die. It means that they're not particularly healthy, trying to make those, because we know we've got different types of enzymes. You'll notice in italic it says porphyrias. They are a group, a family of genetic disorders. They can be altering metabolism in some way. In this case, it's heme formation, which also means you don't transfer enough oxygen around, particularly the peripheral tissue. Now, these are genetic disorders as examples. What's the key bit for you in the report? In the report, you have to identify whatever topic you select in the lab. With that topic, you have to go to KEGG. Remember I mentioned KEGG last week? Big point about that. We'll go through that in the tutorials if you're not so sure. But you need to identify the key enzyme involved in that pathway. What's its EC number? We know what an EC number is, don't we? EC number? Enzyme commission number, okay? You need to know what its EC number is because different enzymes have different commission numbers. Now, in that particular case, you also need to know the substrate in the product, the cofactor. Where do you find that? KEGG. That must be in your report. Is that kind of clear? The one related to what you're doing. For report part B, you'll look at the class of molecule. and I'll talk about those as well. So you need to look at the EC number, what the enzyme is for the report, and we can talk more about that later. So here's an example of a metabolic pathway where things don't work properly, and let's just imagine if you had disruptions to many of those, you probably wouldn't be here. Can I move to the next one? Okay. Probably shouldn't have scared you with the chocolate straight off the bat, should I? Still in shock. Now, I did hint to you that I'd talk a little bit about amino acids being substrates to make nucleotides. Rather than going through every individual detail for each of them, which is rather tedious, I want you to look over on the right and note we're starting with here aspartate. Aspartate, using this carbamyl phosphate, you will see carbamyl phosphate as a name a few times during the semester. What have you just made? Well, you're trying to make this ring structure character here. We have to run through a few series of steps. The hydratase, down here dehydrogenase, incoming NAD+. Remember, that's an energy carrier. But it's not charged up yet. It's getting electrons and protons as a function of this reaction. What did you make? An orotate. Now we've got a ring structure. That ring structure with some PRP substrate, what do we end up with? Starting to look like a base, isn't it? And a sugar coming in. I've circled in red a key character we have to talk about today. Why is it so important? Well, this thing, uridyl, UMP1, which is a monophosphate. It's got a sugar. Just like my laser, write it down. There's the sugar. There's the phosphate hanging off, and there's the base. Base is where we see lots of hydrogen bonding to other bases. You know that. GC, AT, for example. That's got some OH functions, which makes it a little bit labile if it's RNA, for example. We see an ATP coming in with the kinase. What do? You phosphorylate somebody. You're going to activate them. You've got this thing now, uridine 5-triphosphate. So I put a lot of phosphates on. It's active. I've taken glutamine. What have I made? Glutamate. Next step down with this synthetase, what do I end up with? CTP. Now, where do you find CTP? It's got two OHs on that sugar, so it tells me what? Is it DNA or RNA? RNA. It is an important nucleotide. It tells you the functional activity of the cell. If it's making lots of RNA to make lots of protein, the cell is busy. If the cell's not making RNA, it often is going through division, which is DNA replication. Think about the functional activity of the cell. This particular character is important to us. CTP, you see those phosphates on? CTP is also a weak energy carrier. ATP, you know, carries energy, lots of phosphates, 3 of them, high energy phosphates. ATP. Then GTP, next level down energy carrier, still a nucleotide. And then CTP, a very weak one. What you can see here in this little coloured chart from Leninger is that different amino acids contribute different parts to the nucleotide base depending on who it is. That's all I need you to know. There are other contributors. Do you generally make a lot? No, unless you're pregnant making new babies. Repairing tissues maybe. You get a lot in your diet. The other thing that's important here is this creatine phosphocreatine. Remember I mentioned last week creatine and creatinine? Think of them as energy buffers in muscle particularly. They kind of hold the phosphates there ready for fast-acting, hard-working muscles. I'm sitting around doing nothing, suddenly I have to get up and run off. What do I need? Lots of ATP very, very quickly. Takes too long to go through the citric acid cycle. The other thing in italics we're going to see later in the semester particularly is this notion of managing free radicals, bless you. We're going to see a really difficult part of the electron transport chain, making ATP, the last protein I should say in there, using oxygen. When you've got oxygen you can make free radicals. You make free radicals, you damage cells, particularly mitochondria. If you damage them, the cell dies, so do you. What do we have to do? Manage those free radicals. Try and keep them under control so that your mitochondria stay healthy. We use reducing agents like the glycine, the glutamate and cysteine to make glutathione. Just underline it, glutathione. It's a sulfur-based compound that allows you to trap and manage oxygen-free radicals. The other one that's kind of important and less so for this unit is this thing, nitric oxide. For those of you who might be brave enough to do advanced cell biology with me, we come back to nitric oxide. I want you to write down just NO, okay? NO, nitric oxide, not the stuff you put in your cars to go fast or blow the engines up, whatever you decide to do. Nitric oxide is a cell messenger, an extracellular messenger. It's rather unusual. That extracellular messenger is involved in blood flow. It's derived from the urea cycle, which we're going to see shortly. It's derived from epithelial cells that line your blood vessel. It gets a signal, the cells make nitric oxide, the nitric oxide goes from those blood vessel cells, the epithelial cells, to the muscle cells around your blood vessel and change their constriction and relaxation, allowing more blood through. So nitric oxide is often important during a stress response where you want more blood flow, more energy, more work. You increase the diameter of your blood vasculature. It's also involved in clotting. There's a laceration, what do? You set lots of fibrin, there's platelets running in there, you activate everybody. The aim is to reduce blood flow so you don't lose all that blood onto the ground. There's the chocolate and the blood, so it's a good start to the day, isn't it? It's like a horror film. We're comfortable with the notion of how we can use amino acids for a number of things. Extracellular messengers, messengers that do different things, not just hormones like adrenaline, but nitric oxide, an unusual molecule. It is a messenger, but it's unusual. It's just nitrogen and oxygen. Can I move to the next one? Before I do, please just underline UMP, because you're going to see that shortly as a key player of regulating what you do with your urea. So we've started off, Del, a little bit over here. We've made some nucleotides, made some heme, a few other characters as well. We've got an idea, a concept of what they can do. So it's not just making proteins like BSA. We need to get down to this stage now. And I want you to think before we go to this stage, taking in protein. You've ingested it. You had a big lunch, your burger, your tofu burger, or whatever it is, or whatever that new meat product is. I'm trying to find out what it is. That meat product has lots of protein. That tofu has lots of protein. Now we have to manage too much of it, for example. We built things last week, like muscle, etc. We just built a few more things. Now we have to manage the excess. The excess is taking through these characters here, the glutamate, glutamine, the ammonia we're going to manage through this thing, carbamyl phosphate. What's a phosphate tell you? It's busy, ready to do some work. Through this thing, aspartate and arginine, we're going to run around the urea cycle to produce urea. You'll notice the arrow off to ammonia. I mentioned last week, didn't I? There's a small amount in every cell, tiny amounts, tiny amount in your urine. It's when it gets too concentrated it becomes an issue. Can we move to the next one? Yes? Okay. Here's a bit of a summary, just to kind of get you in with your water wings, ready for the deep stuff. We saw last week key players like our glutamate. We highlighted that initially. Here we have amino acids. Put next to that a nitrogen. Whatever's on the carbons, we don't care about at this particular point. We did some last week. What we care about is the nitrogen, so stick it on there. What do? We transaminate. When we do, we've turned alpha-ketoglutarate. Remember that simple molecule, that simple carrier? We then made glutamate. Put the nitrogen on that double dashed line there. You also made these things called keto acids. They're derivatives. They're going to go off and do other jobs. We'll see later on. Now, glutamate can be transaminated back to alpha-ketoglutarate. So draw a line back around to that ketoglutarate there. I've just recycled it. It was just a carrier for a little while, handing off to somebody else. Like a rugby league game. Give it to somebody else to deal with the front. This step carried through this guy, oxaloacetate. What was the acronym I asked you to write down last week? OAA, make sure you write it down again. OAA is contributing to this pathway. Where was OAA? Part of the citric acid cycle. From that, what do we end up with? Aspartate. We can also come in with this glutamate, with the glutamate dehydrogenase. I introduced that to you last week as well, GDH. GDH there is trying to manage the ammonia. There it is coming off. There are a few other reactions we're not going to worry about in this unit right now, but some can produce ammonia as well. That ammonia, that aspartate, what do we end up with? We go around the urea cycle. Now remember, these are roundabouts. Things go in and come out, but the aim is to keep them working all the time. The rate, look at my hands, the rate that turns depends on how many proteins coming in. Lots of proteins, what do? We have to go faster. Less proteins, we go slower. If you're a child going through early adolescence, what do you need to do? Suck out some arginine if you're deficient, so that slows the pathway down again, doesn't it? But as a child eating lots of proteins generally, I know when I was going through adolescence, I was eating more than my father was. Often, particularly as males, you eat more proteins generally. I say typically. So this is a functional where we're going to try and manage that turnover of urea based on supply and demand, what the individual is going through, the rate that we're going to have to turn that. Turn it around to manage ammonia. Do you store amino acids? No, so you have to come through the urea cycle at some point with the nitrogen. Some key bits here. The glutamate collects the nitrogen, that's our carrier, grabs it, keeps it so it's no longer ammonia. Sends it off to maybe make other amino acids, do a few other jobs. It provides one of the nitrogens for the urea synthesis. One nitrogen for urea synthesis. We'll touch on that today as we finish up. The other nitrogen comes from the aspartate, but didn't that come through this convoluted pathway via OAA? Indeed. This nitrogen, some of it, small amount, can be released. A lot of it we want to try and trap. Can I move to the next one? So yes or no? Okay, you can check. So yes? I'm right to go. We're right to go? Okay. All right. You write first and I'll talk about it. What's the first word on the title of that slide? Allosteric. So it's an enzyme. Is it the executive controlling things? Better believe it. Ready to talk about it yet? Yes? Yeah, that's a good response. Well, if you're not on your phone, it might make sense to see what's going on, because this is an allosteric multi-subunit. How many subunits do you see there colour-coded? 6. How many active sites in each subunit? Just 1. Glutamate in, ADP. There's our substrate right next to it, substrate, for this particular enzyme. What's it using as cofactors? ATP, but it's not quite what you think. The ATP we'll see in the next slide is going to help activate and control this particular enzyme. This enzyme is allosteric, and I want you to write down the words relaxed, busy, working. The active sites are open, substrates can get in, it can do its job. But it can also be tense, and you can make this tense by closing those gaps into the active site, and that slows the enzyme down. That's used a lot in biochemistry, enzymology. Relaxed and tense state. Relaxed means busy, tense means not so busy. What does this thing do? Well, you'll see there in rather bold letters, NH4 plus is trapped in this reaction to produce glutamine. You've taken free ammonia with glutamate with this enzyme, glutamine synthetase. I want you to write down the acronym GS. GS. What have you produced? Well, you've used ATP, you've made ATP. There's my glutamine. From glutamine, we can make things, what do you know, like CTP, cytosine triphosphate. We could make AMP as well, adenosine monophosphate. We can make histidine, we can make tryptophan, we can make carbamyl phosphate. Didn't I see that a few minutes ago? Carbamyl phosphate. We can make this thing, glucosamine 6-phosphate, which we'll see in a few weeks' time. We come back to glucose molecules. But the real important thing here is you notice that when these characters, these products from glutamine, did you miss something? You're right. When these characters are made, AMP, tryptophan, carbamyl, CTP, histidine, glucosamine, they're the cars you put on the lot. Right? There they are. If they are purchased, they disappear off the lot, someone's using them. If they're on the lot and building up in numbers, what does that tell your executive about the cars you're making? The Golf, the Tiguan, whatever other things they make, the Beetle. I stopped making that, didn't I? The transporter thing. I've made all those vehicles. If any of them build up, what do you send back to the manufacturer? Slow that pathway down. And I want you to think just like that. If AMP builds up in a cell, it slows this pathway down. Well, why would AMP? Well, let's think about enzyme activity. What do we need to do work? ATP. And write it down on the slide. ATP there up there is being used to grab the ammonia and make glutamine. ATP, the product of that reaction is what? ADP, the diphosphate. Tri, I'm really busy. Di, I'm not so bad. If I take another phosphate off to do some work, what am I down to? A monophosphate. That tells the cell, you had lots of cash for a start, you used it up, I've only got a bit left now. Now I'm going to use up the last of that. I've got no cash left. What does the cell have to do now? If it's down to the bottom of the barrel of its cash, AMP, that AMP builds up. You've got a debt on your credit card, haven't you? What do you have to do? Go and work. Go and work and make more ATP. So whatever this pathway is doing, the cell gets the signal. If AMP builds up in the cell's using lots of ATP, it better start getting busy making more of it. Does that kind of make sense? The same with CTP. I told you that was a weak energy carrier as well. Histidine, if that builds up in the cell, no one uses it. It sends a signal back to this allosteric enzyme, get tense. But don't stop. Now that's kind of critical. We're not going to stop the enzyme at this point. We're going to slow it down. Stop making the cars as fast. Off the production line, slow the rate down. You still do your work, but you slow the pathway down. Glycine and alanine are kind of critical for us. Didn't I mention last week as alanine is one of those common amino acids, particularly in most proteins, not all? Well, here it is here. Now, if it's building up, now what I'd like you to do for each of these characters down here and these two, put a square bracket, concentration, around each of them, put an arrow up. That means concentration builds up in the cell, slows this enzyme down. So we just saw reduced energy available in the cell slows it down, too much tryptophan slows it down, too much carbamyl phosphate, glucosamine, histidine slows it down. But why 2 amino acids? Well, you just saw glycine make heme. Alanine can be used to make what? What did we see last week? Glucose. So if those build up, that means they're not being used to make proteins. Those build up means we need to manage these pathways a little bit more. So these are indicators of the overall, write down that word, overall metabolism of the cell. Now you'll notice down here in italics, I put it down as cumulative allosteric regulation. I've got beetles not selling as fast, so I just slow the factory down a little bit. Oh, I'm not selling the TIG ones. I'll slow the factory down a bit more. This is cumulative. More and more feedback, I start to slow it down. The key bit here is feedback inhibition. Feedback inhibition. Enzymes are getting signals to slow down. And it becomes additive or cumulative. Now that's to manage what you do. Now think about it. I have breakfast at 8, 9 o'clock or whenever you decide to get out of bed. You last another 4 or so hours, you have something to eat, you might have something in the meantime. What do? Running around, do your stuff at the gym and get in the car, come to the lecture, do not much. Look at your phone all the time. See you next year. That way you change your metabolism, don't you? But you change it pretty much like that, don't you? Get up, go for a run. Do you do that? Some people do, some people don't. But your metabolism changes very quickly, so you have to change your metabolism to adjust to what you do very quickly. We comfortable with this concept? This is the first of allosteric regulation. You might think this is complex, but this is the simple one to start with. Can I move to the next one? Anyone got any questions or queries? No? You know it all. We'll be right for the exam, won't we? Yes? Okay. Let's start to dive into the detail of how this works. Now, this is where it starts to mess with your head. I put up again this allosteric enzyme. There it is, ADP, glutamate. There's our 6 subunits all doing work. And now we're going to change the behaviour of each subunit. We're going to do that through this tyrosine linked to position 397 on the subunit. Every subunit has a 397 tyrosine. What can tyrosine happen to it? Phosphorylation, dephosphorylation. Here it is here now. What have I got on it? Not just phosphate, but an adenine. I'm going to change the behaviour of this enzyme by putting this character onto this tyrosine. That tyrosine AMP, and I want you to write in that green box, AMP. What do you see down here? Glutamine synthetase. Look up here, there's glutamine synthetase. Wasn't that the GS I asked you to write down a minute ago? That's GS, same character down here, just not seeing the subunits. Is everyone clear on that? But now what you're seeing is AMP, the guy in the green box. That's why I've colour-coded it just for you. If the AMP is on that tyrosine 397, what happens to our glutamine synthetase? That subunit. It's inactive. I can have 5 others that are still busy, busier, busy less. It depends, but I can activate that one. Does that kind of make sense? So I can actually turn off parts of the enzyme, but not all of it. If I want to activate it, I have to go through what is termed de-adenylation. I'm taking off the green box, this character off the tyrosine, and I'm going to use this phosphate. What am I going to make? An active glutamine synthetase. That subunit. The other 5 are doing their own thing. Now that one's active. Now it can get tense or relaxed, depending on what's happening in the cell. This active enzyme takes what? Glutamate, the ammonia, the ATP, makes ADP, makes glutamine. Let's go back one slide. Isn't that exactly the same as that? I hope so. So I've just made all that. If I want to adenylate, I need to make and use some ATP. This step now helps you regulate each subunit individually. I'm going to inactivate or activate you. We're ready for the next part because I've given you some parts slowly to step you in. Oh, man. I know, it's kind of like that, isn't it? Now, this is where biochemical pathways start to test you, and I say, squeeze your mind a little bit. But it's a good thing to try and push it, to try and fill up your skull, because you know as you age, your brain shrinks. So the more we can fill it up now, the longer you'll last with brain function. In the middle, we see the activity. Inactive AMP with AMP, active glutamine synthetase, it keeps running around. Now, what we're looking at now is the regulators of the regulator. Let's start up here. This character, P2, I'm afraid more acronyms, just its name. As P53, we're going to deal with lots of others. P2 is a protein. That's its name. Bless you, it's a regulatory protein. There it is, that regulatory protein grabbing this thing, adenyl transferase. What's that? ASE tells you it's an enzyme. That's a combination of protein, P2, adenyl transferase. What is that doing? It's grabbing ATP and putting it onto our glutamine synthetase to make this thing. Up here, you should be writing in that green box, AMP again. We're clear on that bit. Let's run around to the next bit. What have we done? We've turned the active subunit inactive. What have we done? We've slowed down this pathway. Let's have a look at the pathway. What's it doing? If the pathway is slowed down, what's happening? We have potentially more ATP, potentially more alpha-ketoglutarate. That's going to alter this character pathway over here, but let's be patient with that pathway. This enzyme can be activated through the deadenylation I talked about. What did you use? Inorganic phosphate. You grab the monophosphate. What did you make? ADP. Who does that work? Adenyl transferase. Except this time, adenyl transferase with its mate, P2. What's P2 got on it? UMP. Where do we see UMP? Let's go back. Where's UMP? Here it is here. Uridyl monophosphate. What is that? A sugar, a base with a phosphate linked on it. That is a key intermediate. What's it doing here? What's it doing here is regulating this adenyl transferase, and it's changing its behaviour. But what I also want you to think about is what it does longer term. What's GS stand for? That's the enzyme, isn't it? The enzyme starts life as a gene, doesn't it? Then mRNA, post-translational modification. There's 6 subunits all doing their work. So if this character is busy turning more enzyme on, your cell's getting the signal that we need to try and manufacture and process more ammonia. What are we doing? Well, that UMP character actually increases gene expression of the GS because your cells are being told you are taking in a lot of protein. You need more of this 6 subunit character to process that. So who would need that, do you think? Somebody on a high protein diet, maybe a carnivore. If you suddenly switch to a low protein diet, what kind of signal would you send to this GS transcription? I'd say the reverse, wouldn't you? Slow down gene expression. Remember, proteins all have turnover rates. They're never there forever. How do you manufacture this guy, this P2UMP? Well, let's come back over here to this pathway. First of all, I want you to look at my laser. Is ATP, put square bracket, arrow up. If it builds up in the cell, what's it doing? It gives you this green triangle. Well, most of you go through a green triangle, don't you? Green means go. It means turn on the reaction, go forward. What's it doing? It's taking, in this particular case, a P2 UTP through this thing, uridyl transferase, uridyl transferase, enzyme transferring the uridyl onto what? Here, onto P2. Just circle P2UMP. P2UMP hooked up with this character says more gene expression. Lots of ATP, what are we doing? Making more GS. Lots of glutamine, if that builds up, what's it saying? Square brackets around it, arrow up. We have a red cross. Some of you on your P plates go through lots of red lights, don't you? But this one's telling you, slow down. Slow that uridylation rate down. Don't make P2UMP. Decrease gene transcription of GS. We're kind of comfortable with this sequence. This is all based on supply and demand. If you're doing economics, we'd have the same kind of scenario to deal with. You've got to think about what these amino acids do. Are they trapping ammonia in this case as nitrogen? Are they transferring on? What's the cell doing? If you're busy working, gym, running, whatever, manual labour, what do you generally do? You eat a lot more, don't you? Carbohydrates for energy production and trying to build more proteins. Protein mass is one of them. But more protein turnover means more pathways like this to manage the ammonia, doesn't it? If you're on a high protein diet and not working, then this should really be running fast, shouldn't it? Yeah? Boy, you thought chocolate was a shock, didn't you? I would suggest to you very strongly that you get to know this pathway. Do you see many real chemical structures up there? No. What you see is names. Who are the key players here? Glutamine synthetase. What are you doing? Managing the transferase of these characters, either phosphates on through this AMP or off. Active, busy. Time for a break? Yep. Might be a good time to duck down the pub, get something to drink, ready for the next session. How are we, gentlemen? Good? How are you? Good? Ladies? Is that comfortable? Get the feeling people are watching over your shoulder? It's like I have that effect on women. Okay.
I just want to know.
Yes, mate.
I was just wanting to know with the.
Okay. Oh, that got your attention, didn't it? He's good with that, isn't he? Oh, really? How about I give you the exam and I'll do it for you? How's that? No. Okay, a couple of questions. People want some clarification, which is fine. Your lab book should reflect the things you do on the day you do it or the pre-work you put in. There's no page maximum. Okay, you write as much as you need to. I'd strongly recommend, given that you've been writing in lab books for a while now, that you write down all of the things you observe. That's kind of important to make sure that you observe things, because that will have an impact, won't it? I asked you to get nutrition labels, but what kind of beverage did you play with your colleagues? Now, I've mentioned to some of your colleagues previously, and I did in the first lab, we're starting this process in the lab of taking it slowly. We're supposed to be focusing on initially what? Specificity. Amino acids, BSA is your standard. We're going to move next week to sensitivity. Have you checked how far, how low can your BSA go to detect things? That's the sort of thing you want to focus on. So we're doing each stage. Look at the milestones and objectives. Look at what specificity does. Look at what sensitivity does. You are scientists in training. These are the things scientists have to go through. You make mistakes, that's no problem. The question I was asked about the team leader presentations, I said at the beginning, we're not assessing your physiology, how well you speak. Some people do it naturally easy because they've been speaking all their lives. Can't shut my daughter up, for example. Even on Skype, so it just doesn't stop. Thank you. Some people make it more difficult, but the idea is to practice. We do not assess that. What we are assessing, if you look at the rubric, are the very simple things, isn't it? Did you get a standard curve? Where are the replicates? Now, I've seen one standard curve that looks more like a relief map of the Himalayas. I won't name names. Is that a problem? Does it show the replicates? That's what the rubric asks for, isn't it? Is that a mistake? Yes. Where was the mistake? Well, let's try and work out where the problems are and solve them. We don't expect you to get it right first time. How many times are you doing these labs? 4 or 5. So you're going to learn about all the issues of assays. Now, as I said at the beginning, these assays, the information you get, the concepts are transferable to any assay. A PCR doesn't matter what it is. It's the same concept. How you deal with things like specificity, sensitivity, interference, Q&As, those things have to be important for any assay. When they do blood samples on you and send you back the results, oh yeah, I accept that, that's fine. My HDL and LDLs are just fine. What do they mean? How did they do it? Those are critical things for you. That's what we're after. So please, we are not assessing your presentation skills. It might look good, but have you got the facts? That's what's important for a start. As I said, as a postgrad, then we start testing whether you can do it more confidently. Comfortable with that bit? Did I answer your question?
PI stands for inorganic phosphate.
That's correct.
And
Yeah.
what's P2?
I didn't mean that question. Did I answer your question about that? Okay. It's okay.
What's P2?
P2 was protein 2. That's its acronym, P2 protein. A bit like P53. P53 is a protein, it's 53 kilodaltons. P53 is an important one controlling cell cycle regulation. P53, if you have mutations, there's whole families that do, they have lots of cancers, lots of tumours. Talk about that in advanced cells. So lots of these proteins are involved in not just what you're seeing here in metabolism, but other functions as well. Should we get back to work? Any other questions before I move on? Okay. I keep talking about this kind of economy. So I want a clarification. Remember I said ADP was kind of one down from ATP. I've lost a phosphate. ATP is the major energy carrier. If I lose another phosphate, I'm down to AMP. That means the cell, if its AMP builds up, there's lots of it, the cell doesn't have the resources, the capability, the energy, the currency to work. So it's going to have to direct its activities to making more ATP. It's kind of like you. If you're busy spending your cash, suddenly you start running out, you haven't got enough for food, what do you have to do? Go and work. That's what you do. Cells got to do the same, except its currency is energy, is ATP primarily. So the cell's making a decision. We've seen anabolism, building things. We just saw a little bit of catabolism. We're breaking nitrogen off, and we're doing that through this pathway. Here's our amino acids ingested. What did you do with them? Well, those amino acids made things. They made GS, they made P2. But those are all turned over. They all have a half-life, which is also critical for your report. If we take the nitrogen off, we're just seeing that a moment ago, we can use that nitrogen to do what? Make other things, if we need to. Things like nucleotides, we saw at the beginning. Some other amines, messengers. From that, we end up with something like carbonyl phosphate. Didn't we see that at the beginning? Carbonyl phosphate, kind of important, activated carbon group in that sense. From that, we might well enter the urea cycle. We haven't seen all the players yet. Those carbons that you've had spare now, what are you going to do? Well, if you've got lots of them, you'll make fat. Some of them might be there to make glucose. A number of them might end up making keto acids. I want you to draw a little arrow off to the right here and put in, once your blood glucose is low, you end up with ketone bodies from those keto acids. Why? Because it takes too long to run around the citric acid cycle to run and make this character, OAA. It takes time. If your brain says you need more glucose, your liver says, okay, I can't supply it fast enough, I'll give you some ketone bodies. What have they done? They're derived from keto acids. We'll see them late in the semester. But what I'm trying to show you here is the citric acid cycle, TCA, Krebs. We haven't seen all the players yet. We know OAA was one of them. If you take OAA out, you can make glucose. But what happens to that cycle if you take OAA out? It slows down. We've just seen a little bit of the urea cycle. I told you it's got to run around and make a few characters as well. What are you trying to make? Urea, excrete that. But these are linked through this oxaloacetate, write it down, OAA, and fumarate. This thing is called the asparto-arginosuccinate. Got to love biochemists with those crazy names. In this case, it's a shunt. I'm taking some things from the citric acid cycle and trying to manage my extra ammonia, too much protein. There's a trade-off of what the cell has to do. I told you already, ammonia is toxic, particularly to neurons, particularly those in your brain. Too much ammonia, what happens to you? We get too much of this going on and we end up with elevated ammonia, tremors, because your neurons can't pump enough sodium out to allow action potential. You get slurred speech. The muscles controlling action in your jaw don't work as properly. They require pumping sodium out. Blurred vision. Well, if you do advanced cell, you see G proteins are involved in sending a photon as a biochemical signal through G proteins, then an action potential back to your brain. Eyes are particularly sensitive. You don't see so well. Then your brain shuts down. Too much ammonia, that's the end of the road. These are critical pathways. And some metabolic disorders, ID, genetic disorders, write it down, might be some of the targets that you have to talk about. So these are some of the pathways that are directly related to assignment part A. But the key bit here is this choice. Not so much choice I want to, choice I have to based on supply and demand. Whether you're going to reuse those amino acids because there's not enough coming in, I have to recycle them as other proteins. Or if there's lots of proteins coming in, what are you going to do? Make fat, write it down on the slide, make some glucose where you need to, but particularly manage the nitrogen as urea. That's the choice the cell has. Which cell's got to make that choice specifically? Which one? Liver indeed, but cell type is hepatocyte, okay? But it is indeed the liver. That's the organ that's doing the work. It's making a decision. Where does that decision get made? What the liver sees as incoming glucose, incoming fat, incoming amino acids. But also the signals from your brain to say, we need to work. We're comfortable with this concept? Yeah? Okay, let's get to the serious stuff. Right, in that grey region there, just look at that first, then I'll let you write. This grey region here is the mitochondria. What I need you to note is those little convolutions, those little bumps in there, the dark grey is the intermembrane space. Here we have the mitochondrial matrix. Most of your cells have mitochondria. They vary in number. They'll do lots of things. That's where you use oxygen. We'll see later in the semester too. But our mitochondria in liver cells, excuse me, hepatocytes, have to manage ammonia, but not all at once. So you write, and then we'll talk a bit about it. And I'll get my exercise walking up and down the stairs. See who's playing on their phone. You realise this is university, you don't have to be here. It's your choice. Myself and my colleagues all have their degrees. It's up to you whether you want one. And as you probably already gathered, I don't give it away. You know you can print. Oh, you've got some. Okay. You ready to talk about this one? Not yet? Like that. Multitasking. Look at me, right. Look at me, right. Are we there yet? Realise all the slides are on the view, so just didn't do them today. It's okay. All right. Can we talk about this now? So yes? What's the cytosol? Close, yep. Where? Yeah, think of it as the cytoplasm, okay? The cytoplasm has lots of cytoskeletons in there, actin, myosin, tubulin, that's for next semester. But that cytosol is kind of more fluid. Think of it as kind of like jelly motion, okay? Things can move around in there. There are cytoskeletal bits like your bones, but things can move around in there. That cytosol, just write down as the cytoplasm of the cell. So we're inside a hepatocyte. We're inside a hepatocyte. That hepatocyte has mitochondria. What's it doing? Well, to manage the urea, we have to take glutamine. You see it here, it's from extrahepatic tissue. What does that mean? Muscle, pancreas, brain, spleen, doesn't matter. It comes from other tissues. Along comes glutamine. It needs to passage through, remember our mitochondria, lipid bilayer, another lipid bilayer, has to get through those. We'll see those are important later on. As glutamine, we have inside there an enzyme, glutaminase. Glutaminase is important because we're going to make glutamine into glutamate. We also see glutamate coming in. Oh, that looks complicated. Amino acids, something like alanine. Didn't we see the alanine cycle last week? That's if you want to make glucose. But when we took the nitrogen off last week, now we have to manage that nitrogen off alanine in the liver. That was busy working muscle. What do? We use this pathway to make glutamate. So you see why glutamate's so important now. Glutamate comes in through our mitochondrial membranes. In here, we've just got lots of glutamate. Now we have to convert that to what? Carbamyl phosphate. Who does that work? This thing. Down next to this, carbamoyl phosphate synthetase 1, write down CPS1. Not just CPU, CPS1. CPS1, grab some bicarb, grab some ATP, lots of it. So now already, to make urea, it's a cost. So even if we're low on cash, we still have to manage nitrogen no matter what. That glutamate is going to donate the ammonia for a short time on our CPS1. You'll notice the bicarb, they've colour-coded the carbon. We're going to use some of that bicarb to help us make this thing, carbamoyl phosphate. There it is, carbon, active phosphate, nitrogen. That's a molecule ready to do something. It's just kind of on raspberry cordial, ready to go to the next phase. The next phase is this reaction with ornithine to make this thing citrulline. Now, citrulline, you think, who cares? Ornithine, who cares? Well, it's part of the citric acid cycle. Where are we? In the mitochondrial matrix. What do you do in the mitochondrial matrix? You turn carbons into energy as ATP. So you're using part of that pathway. Here's the other part of the citric acid cycle, right next to that oxaloacetate, remember? OAA, write it down now so you remember. There's OAA, the conversion there to make this ketoglutarate. What do we end up with? Aspartate. Well, that's kind of freaking me out now. Wasn't aspartate part of the urea cycle we saw earlier on? So here we have the turnover. What we've basically done is taken the nitrogen off an amino acid, the glutamate, the glutamine up here, send it in. We've got a little bit of free ammonia. We've grabbed it with our CPS1. We've made carbamoyl phosphate. Nitrogen on a carbon with a phosphate ready to go to the next phase. The next phase is this thing, ornithine to citrulline. The ornithine looks like that in black normally. See the blue? Comes from all the way up here. Are we comfortable with that step? But you'll notice the arrows egress go outside of the mitochondria now. So you need to jot down the urea cycle. Part of it starts in the mitochondria, that is grabbing the nitrogen, activating it to form carbamoyl phosphate. The next phase, we have to go out of the mitochondrial matrix. Which cells are we in? Hepatocytes. Where are we coming out of? The matrix into the cytoplasm, the cytosol. That's where the work's going to be done in that hepatocyte. Which means this cell, at this point in time, if it's using those amino acids to make our carbamoyl phosphate, aka citrulline, that mitochondria is not doing what as much? It's not making as much ATP. That mitochondria is busy managing ammonia. Can I move to the next one? Sure. All right. Now this pathway is regulated through a series of steps to manage our enzyme production. Down the bottom here, you see bicarb, just look down the bottom, bicarb, ammonia, ATP, carbamoyl phosphate synthetase, what's its acronym? CPS1. Let's go back a slide. Here it is here, down here, ammonia, bicarb, ATP. What are you going to take? The ammonia into carbamoyl phosphate. Here it is doing that, but somebody controls CPS1. Like we saw a while ago with our allosteric enzyme, our GS. Who's controlling this? Well, N-acetyl glutamate. Okay, where the hell did that character come from? N-acetyl glutamate, in this case, comes from glutamate. Wow, this guy shows up everywhere, like a bad penny or gold coin. What do you do with glutamate? Well, you have to activate it. Now, this is one chemical structure you'll have to know. If you've already looked at the slides, you know it's coming at you. What I want you to note is it's acetyl. So those two carbons there, just ring them. Those two carbons there, ring them. That's the acetyl carbons. You're going to see that during the semester, particularly when we deal with fat. Those two carbon acetyls are linked to CoA through the sulfur. Now I said phosphate gets somebody active, so does CoA. Phosphate uses the phosphate high energy electrons in there to help make things busy to do work. Kind of unstabilizes, destabilizes the molecule. CoA does the same, but it's kind of a lower energy. It's not quite as much raspberry cordial. Here it is here, grabbing glutamate, using this enzyme, NAG, I have a few at home, cost me lots of money, N-acetyl glutamate synthase. What's that doing? It's taking glutamate activated acetyl-CoA to make this thing, N-acetyl glutamate. Now you see a green triangle, we'll see lots of those during the semester. What does that tell you about that reaction pathway when arginine gets involved? Speeds up, goes faster. So the rate's increasing. So put a square bracket around arginine with an arrow up. If that increases in the cytoplasm of your hepatocyte, we're going to make more of this N-acetyl glutamate, which is a green arrow to who? CPS1. It says, get busy, go forward. So we've got more going forward in this reaction. What have you made lots of? Carbamoyl phosphate. Now you'll notice the boxed italics. This is critical for the exam. This is an allosteric enzyme. The activation of CPS1 by NAG, think of horses doing something to somebody, get them busy, go faster, exclusively to activate CPS1. So it's just CPS1 we're dealing with now. Now this reaction makes things go faster. Is this the inhibition we saw before with GS? No. This reaction goes faster. More of this pushes this enzyme. You'll notice in the italic on the bottom left, have a look at it. It says feed-forward regulation. Think of donkeys, horses going forward. There's the nag. Feed forward. You have to make them work. You've got to feed them. So you're feeding, you're making the reaction go more and more faster, further forward. So the more ammonia you produce from all the proteins you're eating and not using, turning into fat and sugar, what do you make? Lots of ammonia. Let's think about that. You're on a high protein diet. You're sitting there looking at me. Many of you probably are. So you're making fat, making glycogen, hopefully, making some glucose. What are you doing? You're making lots of ammonia. And if there's lots and lots of it, what do you need? Turn lots and lots of it to urea as quickly as possible. So as soon as the lecture's over, where are you going to duck off to? Toilet, maybe? I won't talk about what these lectures give you, but the aim there is you'll be managing the nitrogen as urea. You're feeding forward. Lots of protein in your diet, not as much work. Don't build protein as body mass. You're turning over nitrogen. Now, this is kind of important for you when you get to later on the semester, after semester break, because we're going to give you a urine, aren't we? I've already asked you to look at its colour. Its colour tells you about what's going on in that urine. What did the individual go through? Are they on lots of water, lots of tea, high protein? Are they urinating blood, as I hinted at already? What's happening to those individuals? Your urine, you should look at it as it's coming out in the morning. Just have a quick look, see what it's like. Is it foaming a lot? What does that tell you? What is foaming in proteins? Seen that, haven't you? BSA, if you shook it up last week, what happened to it? It foamed a lot, didn't it? Not like detergents, it foamed kind of big bubbles and took a long time to disappear, didn't they? BSA is a classic for that. Get a nice dry powder, try and make a solution, shake it up, all these globs of junk and protein and foaming. Takes forever to go in a solution. They are proteins forming bubbles. A hint for you for the lab. So we have a higher rate of turnover in this context because what's driving it? Lots of amino acids. Lots of amino acids, don't use them. Lots of ammonia. What do you need to do? Make more urea to get rid of that waste or toxic product. We comfortable with that concept? Yes? Okay, this red box is critical for us. That's a chemical structure we need to know. Can we go to the next one? Yes? Okay. This is the first one in a handful of key chemical structures. Did I say you need to draw this? No, you need to recognise it. Let's recognise it, shall we? Because the ones I'm going to give you, and I'll tell you now, the last exam question on the 60 or so multiple choice will give you a chemical structure. You have to tell me who it is. Here's the first one, coenzyme A. When I first came to this university, I taught the advanced biochemistry to third years, and I said, well, CoA. Huh? You should know it well before level 3. Let's start down here. Don't write. Look at the board. Pens down. Cross your arms. Sit up straight. Wait for the bell to go. What do you see there? What's that? It's a base. Adenosine. What do you see there? It's a sugar. You see a hydroxyl, there's a phosphate hanging off there. What do you see there? A phosphate bridge, don't you? You're going to see a lot of that, so be careful in the chemical structures I asked you to recall. You then see a bridging group, this pantothenic acid residue. What are you doing? I've got this kind of phosphatey base thing, like I saw with ATP, CTP, with a really long bridge group to who? To this thing, a mercaptoethanolamine. You've all been in chemistry. They show you some beta-mercaptoethanol, have a little whiff of it. Oof, it's a bit pungent, isn't it? It's like rotten eggs. But it has a sulfur. That's the bit I want you to circle with your pen right now. That's the sulfur function. It's kind of like a phosphate. Remember, it has lots of electrons running around in there? That sulfur now is linked to, and I want you to put a box around that, 2 carbons, which is the acetyl. A 2-carbon molecule linked to CoA. That's acetyl-CoA. What does this thing do? Well, it allows you to activate somebody without creating too much heat energy so your cells don't survive. And enzymatically, it means you can manage them into the next step. I've got 2 carbons. Now, while we're here, I want you to note those 2 carbons we're going to see in beta oxidation. We're going to see acetyl-CoA deal with those triacyl glycerols, cut them off, and send them into cells. We're going to see long-chain fats go into your mitochondria with CoA's help. So CoA is seen a lot. That's why you need to know its chemical structure. What it does require, as most of your cell reactions do, not all, requires enzymatic activity. You manage the process. Yes, mate?
Is that whole molecule the pantothenic acid, or is that acetyl-CoA?
Good point. Let's clarify. Okay. This molecule, see my laser running around here? That whole thing is CoA. Okay. There's the base, there's the sugar, there's a phosphate linker. The pantothenic acid is the bridging group from the base sugar to this active end, which is the sulfur. Is that kind of clear? So if I draw a box around CoA and then draw a circular kind of hand drawn around the rest, that is CoA, that's acetyl-CoA. Does that make sense? Must be time to go. Oh, we're still here. Any other queries on this one? As I said, you'll get that chemical structure looking at you in the exam, question 60, and it'll tell me, you've got to tell me which one it is. You're going to get ATP, you're going to get FAD, NADH, and a few others. You'll need to know their chemical structures. They'll look kind of similar, except acetyl-CoA has a sulfur on it, which will make it rather unique, won't it? Can I move to the next one? Is it a yes? Yes. Thank you. Oh, man. Gee. Oh, God. See, I told you you should have gone to the pub, didn't I? What's the top half? Let's go back a slide or two. Isn't that the mitochondrial matrix? What do we have to do? We had to get it out with the help of CoA. Here we are out in the cytoplasm of which cell? Hepatocyte. Now remember, kidney can sometimes do this when it gets desperate, but only when it gets desperate. Out comes citrulline. What do you do outside? You grab some ATP. What do you make this thing? Citrullyl AMP intermediate. Oh, God, they're ugly, aren't they? Base, sugar. See that a lot, don't you? Phosphate linker to what? To this thing. What's the next step? We're going to take aspartate. There's its chemical structure. You know, it's beautifully colour-coded. Gotta love Leninger. Green was the nitrogen. The pink bit is the carbon backbone. Aspartate, what have you just made? AMP. It's cost you an ATP here, 2 ATPs there. We haven't even really made urea yet, have we? So there's 3 ATPs down. we've made this thing, arginosuccinate. So there's the pink carbons from the aspartate, the green nitrogens from the aspartate, the blue nitrogens from back up here in the mitochondria, and the rest of it, which was our carbamyl unit we saw before. We run around to one more step, we've made fumarate, and this thing, arginine. Who needs that? Do you? Is arginine an important amino acid for you, you old people? No. When you were an adolescent, maybe. So just jot that down. There's the arginine of the urea cycle. So that could be used to do other things if you're a child. We take some step with water. What have you just made? Purple box tells you, there's your urea. What we've done now is produced ornithine. Oh, thank goodness for that. Now we've got it recycled back into the mitochondrial matrix and we can use that. So the urea cycle starts where? Well, you could argue it starts outside here to activate it, bring in our glutamines, glutamates, but it actually starts to activate this character, carbamyl phosphate, in the mitochondrial matrix. Where does the urea assay finish? The urea cycle finish? In the cytosol, or the cytoplasm. They're the kind of questions I'm going to ask you in the exam. I'm not going to ask you to draw these chemical structures and steps. Who's the key players? GS, CPS1, NAG, they're the key players. Either inhibition for some pathways, UMP, P2. In this case, we're seeing the production of arginine from, for example, our pathway out of citrulline through here, grabbing how many ATPs did it take to make that urea from the carbon source, glutamine, glutamate. It's expensive. So all that protein as amino acids coming in, you're still going to have some currency. So when your cash is low, you've used up your ATM and you suddenly have to go back to work, you still need money to travel, don't you, to pay your Opal pass. So the cell can't get to zero. That's an important point. It needs ATP, these hepatocytes, just to manage the ammonia you're dealing with. That's the cost for this particular cell as an economy. Our carbamyl synthetase, CPS1, is also related, you'll see, in genetic disorders. You might get something like that to select in the topics I give you in a few weeks' time. It's associated with mental retardation. That's checked when you're first born in this country, because if that's not checked quickly when you first come into the world, because mum was dealing with all that before, once that cord is cut, you're on your own. And if you don't have this CPS1 working properly, what do you have? Brain function doesn't develop. The ammonia screws up all your neurons. So you've got to be treated very quickly. But here's a key hint for the report. I hinted at it last week. Metabolism is kind of amazing because you can find ways around pathway steps. Here are particular steps where we can get around them by using things like benzoate or phenylacetate. I can bypass and help the reaction. They are compounds that help genetic disorders. They are treatments. What are the 4 things you have to do? The research, the diagnostics. How do I diagnose the disease? Well, porphyrias show different clinical signs. Here you might do analysis, an assay, for example. What else have you got? The treatment component. What are you going to use? Well, here you can use compounds to alter metabolic pathway. Now ask yourself, if CPS1 wasn't working too well on you, would you need to be on benzoate or phenylacetate for the whole of your life? Probably. Would you vary the dose if you were male or female, or on a high or a low protein diet, do you think? Or vary the dose through adolescence and then change it when you're an adult? If you're an adult, maybe tissue repair or having babies, should you vary that treatment then? Indeed. And it depends on body mass, concentration. Those are critical components in metabolism. That's a hint for assignment part A and B, that report. Okay. Now it looks ugly, but the key players are pretty simple, aren't they? What would you do? Well, if you're a prudent student looking forward, you'd summarise that. I keep hinting at the key players. How they get to there is what I'm taking you through today. Can I move to the next one? Is that a yes? Or do you want your chocolate now? Can we move on or not? I'm seeing one head nod. I'm seeing, oh, I've had enough for one day. Unfortunately, we have to put this into a bit of perspective. I don't mean perspective in the sense, in relation to the world. I mean in how your cells work. I keep hinting at cells have to make energy as ATP particularly. Here's the character that does most of that work. This character, the citric acid cycle, just put a little arrow down there where my laser's going. Put pyruvate. Put next to it PDH. Key enzyme taking pyruvate into the citric acid cycle. Now this is the first real touch on it. We've talked a bit about it last week. What did I say last week? I said this half of the pathway around this left-hand side, alpha-ketoglutarate, succinyl-CoA. Oh, there's CoA. Succinyl-CoA. So that means it's active. Around to succinate and fumarate, that's where you cut carbon bonds. When you cut carbon bonds, you generate electrons and protons. What does your mitochondria do? It pushes them to the intermembrane space so when they come back in, they make ATP. Remember that ATP synthase, that guy twirling around? That water wheel? That's what you do with the electrons and protons you've just cut out of those carbon bonds. It's where you produce some CO2, the stuff you exhale. Some of that CO2 is going to be helped to make the acid in your stomach. So here we go, alpha-ketoglutarate. What can we start with? Glutamate. What amino acids? Arginine, glutamine, histidine, proline. Succinyl-CoA, isoleucine, methionine, threonine, valine can go into that. Phenylalanine, tyrosine go to fumarate. Run around here. There's our friend OAA. Asparagine, aspartate can be used to make this. Alanine, cysteine, glycine, serine, threonine. There's your pyruvate I asked you to put at the top. That's coming in to make OA. Isn't that going to run around, finish the citric acid cycle? Why are they in brown? Or whatever that colour is you see, pinky brown. Because the carbon skeletons from those amino acids can make glucose. Write it down. Why? Because you're coming around this part of the pathway and go back up through gluconeogenesis. If you're one of these characters in the blue box, like isoleucine, leucine, threonine, tryptophan, tyrosine, tryptophan, oh, tyrosine appears a couple of times. Phenylalanine, lysine, leucine, I can make these characters ketogenic, things like ketone bodies. Now, there's a trade-off here. Think about it. This pathway is a whole series of steps we haven't seen in detail yet, but this pathway we're cutting carbons. It takes time. I could make glucose, but I've got to go back up gluconeogenesis. That takes time. If I'm strapped for cash as energy for my brain, I need ketogenic amino acids. I need ketone bodies. So on this pathway, I can bypass those steps. That's what this particular schematic's showing you. Bypass the steps when the demand for glucose from the brain is high. So write it down, because we're going to come back to that later in the semester with diabetes and other diseases. Comfortable with that overall concept? So now let's think about that BSA you were using in the lab last week. Did you look at the amino acid sequence? How many amino acids in it? What ratio? Which ones? I.e., what concentration? You had a concentration in the lab of amino acids to select from, didn't you? Some of those amino acids can be used to make glucose. Some can't. So if you were on a high protein diet that had lots of these blue characters and not many of those pinky brown ones, what's your poor old brain going to deal with if you're on low glucose as well? Lots of ketone bodies. So your diet, your genetic background, the contribution of what's in your diet, what amino acid composition, will have an impact on what you do, won't it? but also the urea. You have to come back to that. Any amino acids, we have nitrogen. Have to worry about nitrogen on glucose? No. Nitrogen on fats? No. It's just amino acids. Can you store them? No. That means urea's always being made. In that first year human physiology unit, what do they call it now? Not Intro to Human Phys. Sorry? Introduction to Physiology. They make you do the urine prac still. You have to make a pee based on what you drink. I say, oh, you can do it every 10, 15 minutes. You can do it even faster. Why? Because your kidney, your liver is constantly making urea all the time. But you drink more, what happens? Do they do that with you? You increase the volume. Does that increase the amount of urea coming out? No. That depends on what you had a while ago, doesn't it? You just increase the volume, which is different to concentration, isn't it? That's the lab. Volume, how much of the volume versus concentration, how many particles in there. Can we move to the next one? Yes. Everybody else, we ready to go? Okay. All right. You're you, I'm me. Some of you are more male and some more female, depending on your activity levels. Let's look at this. For you, generally, you're producing urea, about 12 to 20 grams of urea on average. Write it down. Average, depending on your average diet. The amount of ammonia, I said there was a little bit in there. Here there is, around 140 to 1,500 milligrams of nitrogen. That's what you're going to excrete every day as urine, on average. Please write down on average, because it depends on what you do, doesn't it? Where you're at. Now, on average, you get a little bit of uric acid. Where do you get that sometimes? Well, the car I borrowed from security has got lots of blobs all over it, going rather brown. But you'll notice this character, creatinine. Didn't I mention that last week? That kind of energy buffer grabs a phosphate, particularly in muscle. It's there ready, so if muscle works really hard, it can supply some ATP very quickly without going through all the pathways we've seen. Creatinine, there's a bit more in males than females. Why would that be the case, do you think? Please write down, indeed you're correct, but generally, okay? If you compared yourself or me to some shot putter female from the Olympics, there'd be no comparison, would there? She's a skinny little thing and I'm all bulked out. That depends on your muscle mass, so jot it down. Now, if that shot putter, male or female, stops exercising, what happens to that muscle mass? Just like everybody else, except there's more skin now, which is mostly collagen, by the way. So here we have the turnover. You have some ammonia, you have some urea, and you have a little bit of uric acid. So you're kind of half mammal, half bird, aren't you? The ratios might vary slightly. But why do these species, particularly birds, reptiles, produce uric acid? See all the nitrogen stuck in that thing? Doesn't that look a bit like a base, doesn't it? Why would they produce that? Well, some of them, yeah. Birds have a slightly higher metabolic rate. They usually run around 41 degrees C. Lots of phosphate turnover, lots of activity. Reptiles, they're trying to conserve water generally, aren't they? So now what they do is they produce this stuff, which you'll already note, if it's dribbling down the windscreen of your car, is more of a paste, isn't it? It's not a solution like urea is in the urine that you've got. You've concentrated. They can manage that. We cannot at high concentrations. That's why we make urea. Otherwise, we'd have white streaks down the toilet bowl, wouldn't we? And that only happens if you've got a particular disease, doesn't it? Are we comfortable with this one? Love toilet human biochemistry, don't we? Okay. Now, it's your job to tell me what's going on in this slide. Read it carefully. Look at the x-axis. What do you have to present in the talk? Team leaders? Tables and graphs. What do you need? Axes, labels. Look at the x-axis. What have you got here? Glucose, hours. Down here it's glucose. Someone's ingesting 700 grams per day. What's that called? The fed state. Someone inducing or taking in 150 grams a day in the fed state. Then we end up here, fasting. When do you start fasting? A few hours, maybe 6, 7, 8. University, sometimes that goes to the middle of the day, depending on the night before, how big a night it was. But if it's lots of booze, then that's going to change the urea, isn't it? More urine. We're fasting for 12 hours, 3 days. Anybody fast 3 days in here? And I mean full 24 hours, not just a day time. Probably not. What about 5 or 6 weeks? Did you know some obese individuals can last a year without eating as long as they get water, vitamins and minerals? We'll talk about that later in the semester. On the y-axis, what do we see? Nitrogen excretions, grams per day. What's that nitrogen as? Let's go back one slide. Ammonia, some urea, or some uric acid. So what's happening in this graph? And a cough's not an answer. I'm listening. Come on, break it down. Problem solve. Have a go. Let's look at the fed state. Let's talk about the fed state first, if you don't mind. Yep. You got the fasting state down, have you?
I was thinking that in the fasting state after time, the amount of nitrogenous waste is held on rather than excreted due to lack of food.
Yeah, you're close, most of it. Yep. But can we go back to the fed state first? So what are we seeing? Urea nitrogen's in the black bar. Other nitrogenous products, amino acids, a few other bits and pieces, are in the white bar. But if you're on a glucose diet, there's your chocolate bar. How much is a chocolate bar, the big ones? My postgrads one that high and then that wide. Took them a couple of months to eat it all. Some university competition. That is 700 grams a day. How much sugar do you eat a day, do you think? Okay, make it easier. How much glucose would you take in a day? How many chocolate bars you eat, isn't it? Other sweets. 700 grams today. Look at that. We're looking at roughly around 2, maybe 3 grams per day of nitrogen coming out as waste product. From what? Well, here we've got glucose that's decreased and the nitrogen coming out is increased. What's that telling me? My glucose intake is high initially. Ammonia coming out, nitrogen is relatively low. Now I reduce my glucose intake and my nitrogen composition, the amount in my urine, is increasing. Why is that?
Because the mitochondria is dedicating more time to dealing with it because there's no glucose.
That's a good point, isn't it? As the glucose levels drop, I spend more of my time in liver cells making more glucose for my brain. You're not in a diabetic state yet, just note that. We're on an average protein diet. What have we got? What's the glucose? Brain's really happy. Glucose drops down, what happens? Hepatocytes make more glucose. More glucose comes from where? Nitrogens as carbons in this case. Remember, the brain needs glucose. It doesn't use fat as energy. Muscles will, but brain doesn't. Who takes priority? Write it down. This does. Doesn't matter what else your muscles do apart from your heart and your intercostals, but this thing takes priority. It will tell your liver cells make more glucose. Okay, now I'm doing okay. I'm down to 150 grams a day. Now you have to fast. No eating. Full stop. 8 hours, 12 hours. We'll talk about interval feeding and fasting later on the semester, how that changes your brain, changes your gut, because your gut can expand, your stomach. You see those guys in those eating competitions, haven't you? I haven't seen too many women. Just shove it down, a plate of hot dogs or burgers. I think it's like, this isn't a TARDIS. It's got to go somewhere. It's going to go in there. But it means your stomach can expand. But your stomach expanding changes what your brain does because it's sending signals to your brain. Now it's full. Well, it was, but it keeps getting bigger. You're fasting for 12 hours. Roughly, that's a good night's sleep, isn't it? So what did you do after a good night's sleep? First thing you do, get up, toilet door, that's mine, don't disturb me. But then if you fast for 3 days, why isn't there more nitrogen coming out in your urine? That's what you had before, isn't it? Because you're starting to run out of those, I won't say excess, but those proteins that you're turning into glucose. You're getting lower and lower. 3 days means you can't just grab them. Now we're going to go out for 5 to 6 weeks, over a month of fasting, no eating, just water, just vitamins, just minerals. What's happening out there? 5 to 6 weeks, what would you look like? More like me. Not much left there. Which means you'll keep the muscles you need the most, which is your heart, your intercostals for breathing, a few other essentials, but anything else, what's happened to it? It's gone. Think of those old war movies, you know, those people who are starving in different camps and the like. They've been fasting for really long periods. They lose protein mass. If you get a broken arm, broken leg, they put a cast on. What happens to it? It starts to shrivel, doesn't it? Take it off. You've got to exercise, lots of physio, to re-encourage protein accretion. Lay down protein. You don't use it, you lose it. Simple as that. But fasting for this long means you're starting to run out of those proteins. Trickling along. By this stage, 3 days, you're already on ketone bodies in the brain. We'll see that in the semester later as well. So should you fast for that kind of time frame, do you think? It'd be unwise. But I'm going to suggest to you there's ways to manage it, to help you manage whatever you want to do. Can I move to the next one? So yes, getting hungry? Thought so. Now, this is also another one for you to think about. Before you write, I want you to just pay attention to what I'm saying. This is an individual. This is another individual. Here's another one down here, another one here. This individual is in a positive nitrogen balance. Growth, who's that? Toddler, adolescence. Growth could be tissue repair. Growth could be more muscle. Growth could be pregnancy. Growth could be lactation. Don't write yet, or I'll turn it off. I'm watching you, Wazowski. So what we're doing here, this process means you're taking in dietary proteins, and lots of those proteins you're turning into tissue, into cells, into enzymes, into things to work. But you remember, you always have turnover. So the ratio means more muscle mass, less turnover, generally, because of what you're doing. A negative nitrogen balance, we're not taking in enough nitrogen, so you see the arrows much smaller, not much going forward. We're pulling back out of that tissue, which we saw in the last slide. In this case, it's inadequate dietary protein. So who gives up first? That's inadequate dietary protein. Down here, it's a negative nitrogen balance because of metabolic stress. What's happening here? A reasonable amount of protein coming in. I am making tissue, but something keeps losing those proteins. There might be a genetic disorder. That's an example for you. On the bottom right, we have a negative nitrogen balance where we lack a specific amino acid. Think again of BSA. Is there anybody missing in BSA? If you only ate BSA, that is blood from a cow, some people do, and it was missing an amino acid, well, corn is a classic. Corn is missing 1 or 2 amino acids, really important ones. You have to supplement. Where's corn used? Mostly in the US. It's a supplement for many of their diets, but there are a few amino acids missing from corn. Your job is to recognise what happens to this overall, depending on whether low protein, high turnover, what your diet's doing, a metabolic disorder, That's the ratio changes. Like I keep saying, depends on the individual. What's the exam going to ask you? What's the status of the individual? Are they a toddler, an adolescent, pregnant female, someone recovering from disease or tissue trauma? What's happening to them? And particularly, and I want you to underline this stuff, the vegetarian. Now, if you're on a strictly vegetarian diet, which is fine, but you need to balance it and supplement in some ways, particularly if you're just on a corn-based diet, because you're not getting all the amino acids you need in that corn-based diet. Trauma is another one. We'll come back to that. That's tissue trauma. I've damaged the tissue, it needs a lot of repair. I don't only need amino acids, I need lots of fats as well to make lipid bilayers of cells, which is critical for me. And these guys, glucocorticoids, what's the one you know about now? What's its other name? Cortisol, indeed, because glucocorticoids is a family. Cortisol, corticosterone, this family of molecules from where? The adrenal cortex. Chronic transcriptional regulators of gene processing. What do glucocorticoids do? Well, unfortunately, they're a little bit complicated. They're a bit biphasic. They generally, though, depending on the individual, induce catabolism. What is catabolism? You're going to lose proteins. Now, we've got to be careful with that because I'm going to talk about anorexia nervosa, which is a brain disorder. That brain disorder is where the pituitary is messing with ACTH. I'll talk about that hormone. Starts in the base of your brain, a little punching bag called the pituitary, the anterior, secretes ACTH, adrenocorticotrophic hormone, peptide. Where does that go into? Your blood runs around, gets down to your adrenal gland, the cortex, and says, release cortisol. But an anorexic person has a dysfunction. They think they're looking in the mirror and they look fat. Brain doesn't recognize that. What do? They keep dumping out glucocorticoids. They keep losing muscle mass. Excuse me. Some of these promote growth. Well, you know testosterone does, doesn't it? If you work out, you would increase muscle mass. And everybody has testosterone, don't they? Yes or no? Yes, including females. And it varies in concentration over time. Can I move to the last one? As I indicate, I'm summarising bits as we go through each of these amino acids. There will be the key concepts or core concepts go up on views after I finish at Hawkesbury, I should say, on Thursday. In summary, amino acid catabolism depending on the variety of cofactors like the THF, the PLP we've seen. They're cofactors. Who are the key enzymes? Who are the key regulators? That's for you to summarise in there. Most mammals, we're focusing mostly on you as humans, ammonia is toxic. You need to capture it. You need to keep it somewhere where it's not toxic. What do? You stick it on our process here and make carbonyl phosphate. Overall, we worked out we're going to use 3 ATPs. That's a cost to the cell as energy, a currency. We're going to use 2 ammonias. Remember, we've got 1 coming in, 1 from somewhere else. We're using CO2. Remember, that was our cofactor there to contribute carbon to make 1 urea molecule. Just make sure it's 1 urea. 3 ATPs for 1 urea. What have you ended up with? Lots of ADP, AMP, and inorganic phosphates. They're a problem. Some of these molecules can be used to make other carbon sources that can be used to make glucose. Things like pyruvate we'll come back to, the ketoglutarate, the CoA, succinate, and OAA. Some more branched chains, the complex ones, make life difficult, so they make ketogenic molecules, ketone bodies. Too complex for us to deal with. I keep hinting at these genetic defects because you have to do a report, because I can't go into every pathway without boring you beyond the current point you're already at and memorising every pathway in that Leninger textbook, which is this thick. But that's no point, because once you get one key example and you've got an idea of feed-forward regulation, feedback inhibition, allosteric, relaxed, intense, a few key players doing this and that, the rest comes easy, I can assure you. We're getting all restless, are we done? So yes? And so it must be time for. That's what we've waited all day for, isn't it? 2 hours of torture just for this. Are we ready? No, yes, no, yes, no. Sorry? Okay. Okay, I agree to that. We'll see about HDs. I don't give them away. Yes, mate.
This is summary 2NH3 plus CO2 plus 3 ATP.
That's correct.
Equal gives urea plus 2 ADP.
Oh, you missed the last bit. Okay. Are we ready for the password? Someone didn't get the full reaction. That's not the password. Password is all caps. Hopefully UMP. I know, some good guesses out there. Good try. Be careful, I keep changing it. It's too quiet, it's making me nervous. UMP. Not ICUP. Okay? UMP. Are we in? Are we in? Good. Just checking. All done? All done?
Yes, thank you.
Can't hear you, still too quiet. Thinking. Tick tock, tick tock, tick tock, tick tock, tick tock, tick tock. See you, ladies. Yes, you do. You do know. Come on, think. See you, gentlemen. Have fun.